and welcome to our presentation on the fascinating world of natural antioxidants. Today, we will be exploring how a technique called paper chromatography helps us discover these vital compounds in medicinal plants. Plants are incredible producers of a vast array of secondary compounds, many of which act as powerful natural antioxidants. These compounds are crucial not just for the plants themselves, but also hold immense value for human health, as well as the food and pharmaceutical industries. Our focus today is on two very important medicinal plants, widely used in traditional medicine, especially in India. The first one is Moringa oleifera, commonly known as the drumstick tree, and Ocimum tiniflorum or holy basil. The study we are discussing aims to compare the phenolic and flavonoid content in these two plants through both quantitative and qualitative analysis. And a key method used for the qualitative aspect, which is what we will dive into, is paper chromatography. This powerful yet simple technique allows us to separate and identify different compounds present in complex plant extracts. So, what exactly is paper chromatography? At its core is a separation technique based on the principle of differential partitioning. Imagine a piece of specialized paper that is our stationary phase. When we apply a tiny spot of our plant extract into this paper and then dip the paper into a solvent, that solvent is our mobile phase. Starts to move up the paper by capillary action. As the solvent moves, it carries the compounds from our extract along with it. However, different compounds have a different affinities for the paper versus the solvent. Compounds that are more soluble in the mobile phase and have less interaction with the stationary phase will travel further up the paper. Conversely, compounds that are less soluble in the mobile phase and interact more strongly in the paper will travel shorter distances. This differential movement results in the separation of individual compounds, appearing as distinct spots on the paper. We quantify this movement using something called the RF value, or retention factor, which is simply the distance traveled by the compound divided by the distance traveled by the solvent front. This unique RF value helps us identify the separated compounds, especially when combined with specific color reactions like exposing them to ammonia fumes, which can enhance their visibility and provide further clues about their identity. Now, Alia will take us through how this was specifically applied in the study.
Thank you, Damya, for setting the stage. Now, let's delve into how paper chromatography was specifically performed in the study to analyze moringa and ocimum plants. The researchers ensured the authenticity of the plant samples by obtaining them from the Botanical Survey of India. The first crucial step was sample preparation. For flavonoid extraction, 5 grams of fresh plant material, specifically the leaves and flowers of both moringa and ocimum, were homogenized. This was done using a solvent mixture of 80% methanol in 1% hydrochloric acid. After homogenization, the mixture was kept in the dark for 24 hours to ensure complete extraction of the compounds. Following this, the extraction was centrifuged at 10,000 rpm for 15 minutes to separate the solid plant material from the liquid extract. The resulting supernatant containing the extracted flavonoids was then concentrated using a rotary vacuum evaporator. Once the sample were prepared, the chromatographic separation began. The concentrated extract were spotted on Whatman number 1 filter paper. The paper chromatography technique used here was ascending chromatography. This means the solvent moves upward through the paper, carrying the separated compounds with it. A specific solvent system was critical for effective separation, a mixture of N-butanol acetic acid and water in a ratio of 4 to 1 to 5. This carefully chosen solvent ensures the differential movement of various flavonoids, leading to their distinct separation on the paper. After the chromatographic run, the flavonoids on the paper were initially detected by observing them under a UV transilluminator. However, for better resolution and to capture clear photographic records, The chromatograms were then exposed to ammonia fumes overnight. This exposure causes a change in the color of certain flavonoid compounds, making them more distinct and easier to visualize. Here, you can see the power of this detection method. On the left, we have the chromatogram before ammonia exposure, and on the right, the same chromatogram after being treated with ammonia. Notice how the spots become much more defined and the colors more vibrant, allowing for clearer identification of the separated compounds. Based on these visible separations and their calculated RF values, the researchers were able to identify several key flavonoids in the plant extracts. For instance, in both moringa and ocimum, they successfully identified biflavonols like chiaflavin with an RF98, various flavins RF73, glycosylflavins RF32, and important flavonols such as myostatin RF47 and chianferol RF84. They also found delphinidin RF45 and various triglycosides RF27. This qualitative analysis confirms the rich flavonoid profile of these plants, which Ayn will now elaborate on in terms of their significance and application.
Thank you, Alia Farisha, for the detailing methodology and results. To conclude our presentation, let's summarize the key findings and discuss their broader significance. The study successfully demonstrated that paper chromatography is an effective qualitative tool for analyzing the phytochemical profile of medicinal plants. Specifically, through paper chromatography, researchers were able to confidently identify various flavonoid compounds present in both Moringa oleifera and Ocimum tenuiflorum. As Alia Farisha mentioned, this included important compounds like biflavonils, flavones, glycosylflavones, and key flavonols such as camphorol and myrcetine. The presence of these diverse flavonoids, along with the high phenolic content also found in the study, strongly indicates the significant natural antioxidant potential of both moringa and ocimum. This is incredibly important because natural antioxidants play a crucial role in combating oxidative stress in the body, which is linked to chronic diseases. Ultimately, the findings from this study, aided by techniques like paper chromatography, reinforce the medicinal value of these plants. It suggests that moringa and especially ocimum, given its slightly higher antioxidant activity observed, could be valuable sources for the development of new nutraceuticals, functional foods, and pharmaceutical products aimed at promoting health and well-being. This research further validates their traditional uses and open doors for the future investigations into their specific compounds. We hope this presentation has provided a clear understanding of how pepper chromatography is utilized in phytochemical research and its contribution to unveiling of plants. Here are the key references that inform our presentation. And thank you for watching.